If everyone could take their seats, please. If you don't have a seat, please find one. We're about to begin. Okay. Good evening, everyone. How are you? All right. How are you? Yes. Welcome to the 17th annual, I guess you can say, Longwood Seminars. Today we are going to be talking about the resistance movement, drugs, bugs, and the fight against Uber germs. Audience members, as well as the social media community, actually had a hand in choosing tonight's topic, and it was one of the most popular, so thank you for doing that. How many of you have attended this seminar before? Oh, wow, quite a few of you. How many is this your first time? Nice, welcome. So glad you came out. So we've also expanded not just beyond these walls here at the News Research Building, but we also have a very thriving social media community who watch this event through live stream. So say hello to the people globally if you want to wave. They can see you. We might not be able to see them, but they can see us. So welcome to all the global community that is tuning in for this presentation. Before we begin, I just want to make a couple of housekeeping announcements. I know, boring, right? So one, I know a good many of people here are interested in the certificate of completion, which if you attend 3 of the 4 seminars, you will receive. PDPs for teachers are available for those that attend all 4 of the seminars. And when you entered in today, you received a question and answer, well, actually a postcard for you to write your questions on. Please feel free. We have staff that will be collecting cards throughout the presentation to answer any questions that you may have. We will be giving them to the panel, and they will address those towards the end of the talk. Videos and reading materials. I know there are a lot of people that were wondering about the supplemental materials that will go to this seminar. They will be available online beginning tomorrow at the website that you see here above. And then also, if you have Twitter fingers, that's a good thing because we are actually having a conversation on Twitter about this. Please visit hashtag HMSMinimed. And then one last thing is if you could put your cell phones on silent, that would be great. It's okay if they vibrate, we just don't want them to ring. Thank you so much. And without further ado, I want to introduce to you our main topic, which is the resistance movement, drugs, bugs, and the fight against Uber germs. With many bacteria becoming impervious to drugs, new approaches are needed to combat the rise of antibiotic resistance. This evening, I'm thrilled to welcome 4 of Harvard Medical School's leading scientists and clinicians to discuss the history of drug resistance, how bacteria becomes able to shrug off the medications we use to treat disease, and ways to address the crisis. I'd like to welcome our first speaker and moderator for this evening to the podium, and it is Mr. Bill Hanaj. He is Associate Professor of Epidemiology at Harvard University and the T.H. Chan School of Public Health, and is also a faculty member here in the Center for Communicable Disease Dynamics. Bill?
Thank you.
Thank you.
Thank you so much, Mary. Thank you, and also thank you all, because you selected this. This is a very important and very, very, very important topic, as I think we're going to cover over the next hour or so. Could I just get started by asking you to reach over? I know this is going to be a little like a revivalist meeting, but if you could reach over and shake hands with the person next to you. This is not an attempt to get you to transmit disease. And then it'll all become clear later. And then on the other side as well. Yeah, look, yeah, that's it. You guys as well. Thank you. Great. This is all going to become clear later. This is not. And if you're watching on the internet, shake hands with whoever's nearby. You know. So I want to start by asking, what would a world without antibiotics look like? What are antibiotics? Well, they are the drugs that we use to treat bacterial infections. Bluntly, they kill bacteria, or at least stop them growing long enough so that the immune system can kill the bacteria. So they were first discovered in the 1930s. Famously, penicillin was found by Fleming. We'll come back to him later. It was only actually developed as a sort of useful therapeutic during the Second World War by Florey and Chain. But since then, people have estimated, and I don't like coming up with big numbers off the top of my head because there's a lot of uncertainty in them, but people have estimated that penicillin has saved 200 million lives. Regardless of whether that's exactly correct, it's a big number. So a lot of people think that losing antibiotics would take us back to something like this. This is a Bruegel painting called The Triumph of Death, and it's based upon the catastrophic epidemics of Yersinia pestis, plague, which bedeviled Europe in the pre-modern era. They could take away a third of the population. And a lot of people think that, and it's readily treatable with antibiotics, a lot of people think that that's what losing antibiotics would be like. But actually, I think it's worth thinking a little bit more carefully about what the value of antibiotics is to modern medicine. And it's not so much that it's fighting the Black Death, is that it makes so many things in modern medicine possible. For instance, whoa, I was the first person to make a mistake with this. There we go. Transplant surgery or cancer therapy frequently require that we suppress the immune system. And if we suppress the immune system, that leaves the body prey to bacterial infections. If we cannot treat those bacterial infections, people run the risk of very serious consequences, death, bluntly. Basic procedures or basic things that we might not have thought about. I mean, a burst appendix, to be sure, is not something that you want to happen, even now, because it will let loose a deluge of bacteria into your body that you don't really want to be in your body. You want them in your appendix. If they are resistant to antibiotics, then we're going to be unable to treat that, and that could become deadly. And then there are the things that we find in the community, the pneumonias, the gonorrheas, and the tuberculosis. Tuberculosis is something that one of our later speakers is going to cover in depth. As these become untreatable, then we run the risk of being able to pick up diseases within the community that could kill us or leave us infertile, or many other negative consequences. So, just to put some numbers on it. A few years ago, I was asked with others to, I went to a meeting at the World Health Organization, and they said, what's the burden of antibiotic resistance? And as I said earlier, it's very hard to come up with accurate numbers, but the numbers are large. We estimate that worldwide, this is from the Wellcome Trust, around 700,000 deaths occur every year from antibiotic resistance, around now, this is. In the United States, there are about 2 million serious antibiotic-resistant infections every year, and about 23,000 deaths. To put that into context, somebody will die while I am talking to you now, probably, from an antibiotic-resistant infection. But that's now. And as we've said, this is a growing problem. Some people have tried to estimate, tried to run the tape forward to see what the situation could be like in 2050. In 2050, we're looking at a worldwide annual death toll of around 10 million. To put it into context, the 1918-19 flu pandemic killed about 40 or 50 million. So it's not up there, but this would be an ongoing thing, not a pandemic which went away and came and didn't come back the next year. The United States, over 300,000 deaths. And these numbers, this is a caveat, these numbers were not estimated by an epidemiologist, which is at least what I'm a professor of. They were estimated by an economist. And I don't want to be mean to economists, but they're not epidemiologists. Regardless, these are very big numbers. They're things which sort of put, this is why people like me are concerned about it. And no matter, I hate to quote Stalin, but he said that, 1 million deaths is a statistic. A single one is a tragedy. Each one of these would be a tragedy. And I'm sure many of you read in January about the lady in Nevada who came back from overseas with an antibiotic-resistant blood infection. And it was resistant not just to the frontline antibiotics, it was resistant to all 26 that were used to attempt to treat her. And she died. And those are rare, those infections, and still for now. But just to, before you get too comfortable, this is what we call an antibiogram, or the susceptibility data for an isolate that came from a hospital in the Boston area. And these are the drugs that we would think about treating with, and you can see resistant, intermediate. Intermediate still isn't good. It still means not fully susceptible. So this is a problem which is not remote. It's coming closer to our door, and we should be thinking about doing something about it. So, what makes resistance happen? Why does antibiotic resistance exist at all? And I'm assuming that many of you will know the answer to this, but it's worth reiterating. Bugs are resistant to antibiotic resistance. Bugs are resistant to antibiotics because of the same reason that peacocks have tails. Because peacocks with prettier tails leave more descendants. And in the same way, cheetahs that can run faster are more likely to catch the gazelle, and they're going to be able to feed their little cheetahs. The gazelles that can run faster are more likely to outpace the cheetah and survive to live another day and have baby gazelles. Because those properties are inherited, natural selection means that these two things are very, very fast, and that this is one of the great battles of nature. In the same way, bacteria that are not killed by the antibiotic survive. They live to fight another day. They proliferate in the population until we have a problem with antibiotic resistance. It's evolution. So this was something that was actually, you would think, since we have known about evolution for over 150 years, that somebody would have thought about this. And indeed, they did. Alexander Fleming, in 1945, when he was about to be awarded the Nobel Prize in medicine with Florey and Chain, who actually did the hard work of making it into a proper concern, a proper antibiotic you could use in medicine. He gave this interview to the New York Times, and I will leave you to read it. You can't quite see it. The microbes are educated to resist penicillin. I think you can still get the gist. He makes here that the thoughtless person playing with penicillin is morally responsible for the death of the man who finally succumbs to infection with a penicillin-resistant organism. He predicted it. He knew it would happen, and within years of penicillin being introduced and used in medicine, it did happen. Fleming discovered penicillin when a Penicillium notatum, which is a mold, a spore of that mold floated through the window of his lab, which was in the building opposite where I used to work, and landed on a plate of Staphylococcus bacteria, like these. And it grew there. And it was able to grow there because Fleming had gone away on vacation, and he had not bothered to clear his bench. But when he came back, he saw that it had grown, and these are actual discs of paper containing antibiotics, so you can see it, but he found around it this perfect zone of resistance, zone of inhibition, where the bacteria were unable to grow. And that was how he first discovered the effect that would lead to the development of penicillin. I want you to remember that, because it's a naturally occurring molecule. We'll come back to that in a bit. So, the raw material for evolution by natural selection is mutation. And this illustrates a mutation. DNA double helix. The A's and T's and G's and C's, which we all know are nucleotides, are just shown as colors here. These are identical, these two DNA strands, apart from this one, which has generated a single nucleotide polymorphism, one mutation. Mutations do not happen often. But when it comes to drug resistance, they happen often enough. There are some classes of drugs and some resistances for which a single mutation is sufficient. And that might seem unlikely, but you have to remember that within each of these are several different types of bacteria growing on a plate. Within each of these colonies, there are millions of cells. Now, what you have to remember there is that even if the mutation is a 1 in a million chance, these things are rolling the dice a million times. So the population size of these things is such that they have explored a lot of the evolutionary space already, even before we get to it. You have around 1 billion E. coli in each of your guts, plus a whole bunch of other things. So you may be wondering, with some justification, why I've put a Sudoku up in the corner. But one way that I put this across is that, this is a relatively difficult problem, okay? If I were to give this to one person in this room, I'm sure, because you're all very good at Sudoku, that it would be solved quite quickly. But if I really wanted to get it solved quickly, I would take it to Fenway Park, I would give it to everybody in the entire ballpark, and I would say that the game is not going to start until somebody has solved it. And somebody, I'm sure, would solve it pretty quick. I would be sampling a wider range of people, I'd be sampling far more of them, and I would be motivating them as well. That's the thing with mutation. I now want to tell you what I think is probably the most interesting thing I know that is not widely appreciated by the public, which is about the mobility of DNA in bacteria. What do I mean by mobility? I mean that in bacteria, genes do not stay within a single lineage. They can move from one to the other. Sometimes they're on things called plasmids. Sometimes they can be moved by viruses. Other times, bacteria will just take up DNA from their environment, start making use of it, which means that rare traits can spread. So just to illustrate this, if you play a musical instrument of any kind, can you raise your hand? You don't have to play it well. I'm not going to ask you to play it. Okay? Quite a lot of people. You are a sophisticated crowd. Okay. Now let's ascribe a fitness value to that musical instrument playing. Imagine that we were only feeding you if you could sing for your supper, and everybody else would not be getting any food. It would become a very good thing to be able to play a musical instrument, and so you guys would have an advantage. But just imagine if that ability to do something complicated, like playing a musical instrument, could be passed on to a neighbor by something as simple as shaking hands. Raise your hands now if you shook hands with somebody who played a musical instrument. You see? Suddenly that ability is way more widespread than it was previously. And natural selection has a lot more to be able to work with. And bacteria do not only do this within a species. No, they do not. Look at this. This comes from some work that I did, in fact, with Dr. Kirby, our second speaker. He was also on this paper. This shows family trees of 3 important bacteria, Enterobacteria cloacae, E. coli, and Klebsiella. These all came from the Boston area. What you need to know is that these brightly colored things link things where exactly the same gene was found in different things. I mean, exactly the same gene. So here you can see that this, which is a Klebsiella pneumoniae, is also found in E. coli, which is a different species. To put it into context, this is as if you were to be bitten by a spider and gained the ability to climb on walls. And bacteria don't even need to be radioactive. It could be. So I need to walk over here to remind myself what the next slide is now. In closing, I want to ask the question of, I've talked about how natural selection has worked in the antibiotic era. Does this mean that evolution has just worked really quickly? Well, no, it's not so much that. There are so many antibiotic resistance genes because antibiotics are everywhere. This, again, is from a piece of work I was involved in. This is a piece of permafrost core from Canada. So this is from the tundra. As you dig deeper and deeper and deeper and deeper, this is stuff that was laid down longer and longer and longer ago. So bacteria fall from the atmosphere where they get carried by upcurrents and blah, 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 fall and they get frozen. So you have a deep freeze, and as you go deeper and deeper and deeper into it, you're reading back in time what was living then. We found a gene existing in a Staphylococcus, it looked like, about 6,000 years ago, that was when it was laid down, that layer within the ice core, that was capable of conferring resistance to this. This is amikacin. Amikacin is what's called a semisynthetic antibiotic, which has never existed in nature. It was only developed in the 1970s. So what was something doing 6,000 years ago? It's not that it was resistant to amikacin. It was resistant to something that was a bit like amikacin. Remember, Fleming found the antibiotics as part of a natural process. These are natural molecules which have been made in nature for millions, billions of years. In fact, when I said imagine a world without antibiotics, there has not been a world without antibiotics for millions of years, not since bacteria and fungi first started their arms race, whenever that was. I'm sorry, I'm not that kind of biologist. I can't think that far back in time. There has never been a world without antibiotics. And so we are continuously playing catch up. And we, in our medical attempts to prevent the hideous consequences of bacterial infections, are continuously having to deal with the consequences of this ancient story. So that was my introduction, and I'm now going to hand over to the first of our three marvelous speakers who are going to take us through the topics which I've put up here, and then we're going to come back and answer all of your questions. And I want to thank them for speaking next. I want to thank all of you for listening and for coming. Thank you.
Okay, as Dr. Hanage was just telling us, antibiotics come from the environment. There's warfare going on in the environment between different microbes that are trying to stake out territory, and there's a limited resources and food sources. So microbes have developed the ability to produce antimicrobials, antibiotics to kill off other organisms in the environment. And we have co-opted those antibiotics to use in treating infectious diseases. And so I wanted to introduce you to the players. And so we have us, human cells, and bacteria. And a human cell, if simplistically, is a bunch of protoplasm surrounded by a cell membrane. And bacteria also are protoplasm surrounded by cell membrane, but they differ from us in that they also have a rigid cell wall, shown in gray. And there are some bacteria, not only do they have protoplasm, cell membrane, and a cell wall, but they actually have an extracellular membrane. So bacteria look a little bit different than we are, and those differences can be exploited and targeted selectively by antibiotics. Now, in terms of the toughness scale, it's really easy to kill human cells. But bacteria have all these extra layers, and it's kind of hard to kill bacteria. And it's really hard to kill these gram-negative bacteria because they have all these different layers. So the trick is to find things that can kill bacteria, but not us. And microbes produce a lot of different things. They produce compounds which can kill human cells too. So, we've gone and looked for things that are very selective for bacteria, and those are the things that we're using in human therapy. And I just wanted to show you that antibiotics, in order to get into bacterial cells and have this very selective ability to kill bacteria, they turn out to be these really super weird molecules. So if you look at, for example, a molecule or a compound that we use to treat human diseases, non-infectious diseases, that just have to get into our own cells and not bacteria, they tend to be pretty, pressing the wrong buttons, they tend to be very simple molecules. Okay, the structure's not important. This is an example of a common ulcer medication called Nexium or esomeprazole. This is what a typical antibiotic would look like. A chemist in his or her wildest dreams couldn't think up this structure. It's very difficult to synthesize, but microbes over millions and millions of years of evolution have figured out just what's needed to get past bacterial defenses and kill other organisms. Okay. So bacteria, because of the structure that they have a fundamental problem. And so bacteria are surrounded by a rigid shell. So if you're surrounded by a rigid shell and you need to expand and grow, you have a problem. You can't really stretch that shell out. And so the solution that bacteria have is what I call the rigid shell paradox. In order to grow, you actually have to snip that shell. So what the bacteria do is they snip the shell in multiple places, and then they stretch out a little bit. And then if they kept on cutting and stretching, the cell wall would fall apart. So what they do is they stitch the shell back together. So they cut, expand, sew back together. So now I wanted to introduce you to our first antibiotic, penicillin, that Dr. Hanage was referring to before. And so what penicillin does is it binds to the stitching mechanism. So the bacteria can no longer sew itself back up together. And so what happens then, and I'll show you an example. These are bacteria under a microscope. These are Escherichia coli, a bacteria that's found in our guts. And you describe them as short, rod-shaped organisms. And now if we look at those organisms and expose them to something that inhibits the sewing machine, in this case a cephalosporin, it's very similar to penicillin, what happens is the organism keeps on cutting and it doesn't stitch things back together again. And so the organism literally falls apart and it starts to swell up, and you can see these bulges and they form these long filaments, and this is incompatible with bacterial life, so the bacteria are killed. Okay, so that's an example of how one major class of antibiotics work. Now, there are a number of different targets in the bacterial cell, and I'll just briefly go through some of them. So some of them attack these special bacterial membranes that look different than our own membrane in this cell wall. Penicillin, we've just spoken about. And then bacterial cells, just like we do, have a genetic blueprint, and there are antibiotics like fluoroquinolones that prevent replication of that genetic blueprint. And then that genetic blueprint sends out instructions into the cell to tell the cell what it should make. And those instructions are messenger RNA, and there are antibiotics that interfere with the production of messenger RNA. And those instructions go to the manufacturing centers of the cell, which are called ribosomes. And those ribosomes make the structural components of the cells, the enzymes that carry out various functions in the cell. And these manufacturing centers, ribosomes, we have ribosomes too in our cells, but they look very different. Bacterial ribosomes look very different than our own ribosomes, so that there are a number of different antibiotics that selectively target this machinery. Okay? So those are examples of the major types of antibiotics and how they can inhibit bacteria. Okay, so I'll bring you back to the soil, my environmental slide. And the red bacteria have been busy killing off the purple bacteria. But as Dr. Hanage was saying before, evolution's going on. And eventually, one of the purple bacteria figures out a way to resist that antibiotic. And so there are several mechanisms by which this can occur. And so if we go back to our sewing machine example, we have another a molecule called a carbapenem or meropenem that can bind the sewing machine. But now we have an organism that has acquired or developed a resistance mechanism. So one way to resist this antibiotic is to break the antibiotics down. So there is an enzyme which will cut the antibiotic, and that changes its shape and function, so they can no longer bind to the sewing machine. So this organism is now completely resistant to penicillin. Well, there are a number of different ways that organisms can become resistant to antibiotics. Another way is to not cut the molecule, but you can use a molecular staple to staple a chemical group onto this antibiotic. And when you do that, the antibiotic looks different, okay? So it has this big bulge over here, so it can't bind its target anymore. And yet another way is you can actually change the target itself through some mutations. So the shape of the sewing machine is different. So this, for example, penicillin can no longer bind. Okay, I want to bring you back to this gram-negative bacteria, these really tough bacteria that have these two membranes. And so it turns out that bacteria also have to get nutrients through these two membranes too. So in their outer membrane, they have these pretty large pores or holes. They're called porins, so that nutrients can flow into the bacteria itself. And so antibiotics can also flow in through those pores and then access the cell wall or get into the cytoplasm. So what bacteria have also figured out how to do is they can acquire mutations in those pores. So those pores suddenly become smaller. Okay? They're too small for the antibiotics to come in, but their nutrients can get in well enough. Maybe not perfectly, but now the organisms are again resistant to this antibiotic. And if the antibiotic still gets in, the organisms have acquired or developed sort of pumps. They're called efflux pumps, and they can pump the antibiotics out. So you can see that there are multiple different ways that organisms can become resistant to antibiotics, and sometimes we find multiple different mechanisms in the same strain. Okay, and as Dr. Hennig was saying, one of the most remarkable things is he showed when we shook hands with each other is that antibiotic resistance can be fairly easily transferred from bacteria to bacteria. And antibiotic resistance genes are often carried on these autonomously replicating pieces of DNA. They're not part of the bacterial chromosome. They're these circular pieces of DNA called plasmids. And often these plasmids encode multiple antibiotic resistance genes, resistance not to just a single antibiotic. In the study that Dr. Hennig was referring to, some of these plasmids maybe have 5 or 10 antibiotic resistance genes. And in addition to encoding resistance genes, they encode a machinery that allows them to transfer the plasmid, allows the plasmid to transfer itself, to replicate, and transfer itself to another bacteria. So this is a molecular mechanism. It linearizes itself, and then it forms a plasmid in an adjacent bacteria. And what's even more remarkable about these plasmids is they also often encode an addiction machinery. So basically, if a bacteria is foolish enough to try to kick out this plasmid, the plasmid ends up killing the bacteria. So the plasmid encodes a very stable toxin and an antidote to that toxin, which is very short-lived. So if the plasmid happens to be lost, then the toxin hangs around, the antidote is gone, and the bacteria is killed. So now this purple bacteria has transferred its resistance to other bacteria, and we have this poor red bacteria, which used to seem to be doing pretty well for quite a long time, and so it's stuck. And so now its goal is to acquire some new antibiotic. And what happens is, what is happening is a antibiotic arms race, if you will. And I'm showing you one class of antibiotics that attacks the bacterial cell wall, like penicillin that I told you about. And in terms of human therapy, we've started out using penicillin and ampicillin. Organisms had developed enzymes that would cut up those antibiotics. And then we went into the soil and we pulled out first and second and third generation cephalosporins, and then the pathogens acquired, antibiotics that could now, enzymes that can now break down those resistance mechanisms. When I was in medical school, we used to think about this wonder drug called meropenem or carbapenems, which could basically kill any bacteria that you could think of. And recently, there's come into our bacterial populations enzymes that can degrade these antibiotics of last resort called carbapenemases. And so there are these carbapenem-resistant Enterobacteriaceae. The CDC is calling this one of our most urgent antimicrobial resistance threats because they're resistant to this, what was a fairly safe, non-toxic antimicrobial that had sort of been one of our last resort medications against these very difficult to treat gram-negative organisms. Okay, I wanted to bring it back to our patients. And so in our patients, they may develop an infection, and we treat them with antibiotics, and the infection's cured. But during that process, remember, antibiotics don't discriminate between pathogens in our normal flora. So our bodies are covered with bacteria. We have bacteria in our upper respiratory tract and in our intestines. So the antibiotics kill that normal flora too. And in our hospitalized patients especially, this sets up a vicious cycle of resistance. So we treat our patients, their normal flora is knocked out, so then they become colonized with organisms that are resistant to the antibiotic we just treated our patients with. And especially in our hospitalized patients, patients with a compromised immune system, who might be on chemotherapy, who might have had a transplant, those patients get infected often from their endogenous flora, their colonizing flora. So now they become infected with a pathogen that's resistant to the antibiotic they were just treated with. And then they become colonized with more resistant bacteria. And this cycle goes around and around until the patients may get colonized with an organism such as Clostridium difficile, which produces a toxin which causes a very severe colitis, which can be lethal. Or they're infected with one of those organisms, Dr. Hanage showed an example of which are either resistant to all available therapies or effectively resistant to all therapies, or only toxic therapeutics remain, and that leads to adverse outcome. So, in summary, antibiotics and resistance elements are borrowed from an ongoing bacterial arms race, and bacteria are very difficult targets. It's really difficult for chemists to synthesize new antibiotics. We generally pull them from the environment and where the bacteria have done most of the developmental work for us. There are many, many paths to antimicrobial resistance. We amplify resistance in our hospitalized patients. So you have to think about careful use of those antibiotics. In escalating antibiotic resistance is a looming societal problem. So with that, I would thank you, and I'd like to introduce our next speaker, Dr. Farhat.
So, make sure this is working, perfect. So I'm going to talk to you a little bit about the global impact of drug resistance. And I'm going to use specifically one example of one disease, that's tuberculosis. And for many of you haven't seen a tuberculosis case in your life. It seems like a kind of a remote disease that affects people across maybe different seas, or really is not a real problem anymore. But I'm going to, in the next few minutes, I'm going to try to convince you that, in fact, it's very much still a big problem globally, and actually one of the major types of infections that drug resistance is hitting the hardest. So back in 1812, so about 200 years ago, in Massachusetts, about 1 out of 4 people died from tuberculosis infections. So move along to 2017, we barely have any TB in the US. Only about 3 cases in 100,000, and almost none of them die. And all of this is, thanks to antibiotics. And I think many of you have used antibiotics in your day-to-day. TB is a particularly challenging infection to treat because you not only have to take one antibiotic for a week or 10 days. Typically, you have to take 4 or sometimes more antibiotics for as long as 6 months in order to get cured. So this is a particularly big challenge. So the fact that we were able to get this far in the US and Europe and many parts of the developing world is really a great success story, and really is a story that we need to protect and a reality that we need to protect with everything we have. So in 2017, we have the latest statistics from the World Health Organization tell us that we still have over 10 million people fall ill with tuberculosis every year. In addition to that, tuberculosis alone is actually the top cause of death for any single infectious organism, more than malaria and HIV combined. So, and I think in addition to the fact that, in fact, most of these cases are happening overseas. Most of them are in sub-Saharan Africa, Southeast Asia, so it's particularly India, China. However, our world is no longer like it used to be. Their borders are not really real. And in fact, people are constantly traveling across the world. And this is really a reminder that, in fact, in 2017, what's happening across the world or across the ocean is still should be relevant and should be a concern to us here in the United States. And one of the major challenges that's really threatening TB control, tuberculosis control worldwide, is this emergence of drug resistance. As I mentioned before, treating a regular or drug-sensitive TB infection takes us 6 months. But treating a drug-resistant infection can take us as long as 2 years and sometimes longer. And there are, So this is sort of a map showing you that, in fact, drug resistance around the world is not evenly distributed. It tends to be, so darker here means more cases. And in fact, it is more common in, for example, the former USSR region, Eastern Europe, and some parts of Africa. But it is still something, almost every country in the world has noted and reported drug-resistant TB cases. And this is a little diagram to remind us that not only can drug resistance develop in one person, this is as a result of evolution, as previous speakers had mentioned, when the organism is when you're treating somebody with an antibiotic and they're taking the medicine, but then the organism evolves within their body to develop the drug-resistant mechanism. So this is the so-called primary resistance, when you have perhaps they didn't absorb the drug, perhaps they missed some doses, and then they acquired resistance. But increasingly what we're seeing is not this acquired acquired resistance phenomenon. Increasingly, people who have never seen the infection before are getting infected by others who already have that resistant infection, so meaning they're transmitting. These resistant infections are now being transmitted from one person to the other. And this is a major cause of concern when you have done everything right, so to speak. You've taken your antibiotics, but just because you're in the wrong place at the wrong time, you end up getting a drug-resistant infection. So, and this is from NPR a couple of years ago, where in India, they've noted the so-called totally drug-resistant TB that was essentially resistant to a panel of the 13 drugs that are currently used for TB therapy, which was a major cause of alarm. And in fact, a lot of people were saying that this is threatening to take us back to the era where all we could do for tuberculosis is put somebody in a sanatorium and pray. So what is the major problem with drug resistance in tuberculosis? So as I mentioned, it's particularly challenging to treat. And so in the best hands, it can take up to 2 years of fairly toxic therapy. But the reality is, in the majority of cases, if you imagine this is sort of a sea of patients that all have multi-drug resistant TB, which is TB that's resistant to rifampicin and isoniazid, 2 of the most potent drugs that we use to kill it. The reality is only 1 out of 2 of these patients get diagnosed. That they eventually get recognized and they undergo some kind of testing that proves, in fact, that they have drug resistance. And even then, 2 out of 3 people who actually are diagnosed eventually get access to treatment because treatment is expensive and it's limited. And so a lot of people don't actually get treated even after they're recognized to have drug resistance. And even after people go on treatment, in fact, because the treatment is toxic and because these people tend to be fairly sick, only 1 out of 2 of those patients get cured of their infection. So we're currently in this dismal situation of only 1 out of 6 patients with drug-resistant tuberculosis gets cured of their infection, and 5 out of 6 go around potentially spreading resistance in their communities and really threatening the public. So this is a diagram from the Centers for Disease Control, just to demonstrate that, in fact, how expensive it is and the toll that it takes to have a drug-resistant infection relative to having a regular or drug-sensitive infection. So you can see that, these are estimates from the United States, obviously, so these numbers are, much higher than they would be, say, in sub-Saharan Africa, but nevertheless, you get the idea that it costs a lot to actually treat, the cost of medicines, the cost of the physician, the cost of the follow-up. But in addition to that, the loss of productivity, because these infections, especially tuberculosis, tends to affect young people in their prime years of life. So there is considerable loss of productivity. And in fact, it is among the top causes of kind of bankruptcy or financial ruin worldwide. So why is tuberculosis drug resistance and tuberculosis particularly challenging to diagnose? And the reason is because, in fact, contrary to some of the other infections we're used to, like, strep or urine infection, so the things that are more common, TB is a very so-called slow-growing organism. So when you try to take a sample, say somebody coughs into a cup, and you try to take that sample to try to identify whether or not it contains TB, typically what you're doing, you're putting it on a little Petri dish, and you're watching for growth of the organism. And in fact, in most cases, it takes about, for regular infections, it could take about a day or 2 or maybe 3 for the bacteria to grow. But in the case of TB, it could take weeks. 2 or 3 weeks for you to recognize, in fact, that this is tuberculosis. Most other tests that we have at the moment are poorly sensitive, don't tell you reliably that you have that infection. And then once you have the culture telling you that, in fact, you have TB, you then have to grow it in the presence and absence of the antibiotic that you're going to use for treatment to see if it's, in fact, sensitive to, so similar to the plate that you saw with Dr. Hanage earlier, where you had the little mold, and you essentially try to see whether or not it's going to grow around that little pill of antibiotic that you put in there, and essentially try to assess whether or not it's sensitive. And so based on that information, usually clinicians decide whether or not to give the patient that antibiotic. And again, that takes several weeks. So you're talking about a delay of 1 to 3 months in the case of tuberculosis before you're able to essentially design a treatment regimen for the patient that they're going to respond to. So this is obviously a big problem. So how do you get around this issue of needing to do culture? So as you heard before, drug resistance is so-called heritable. It's passed on from one bacteria to its offspring, because it's heritable, it's encoded for in the DNA of the organism. And so instead of trying to grow the organism and test it in a Petri dish, what you could try to do is read its DNA and try to predict ahead of time identify those mutations that are causing the resistance, and then make that determination right then and there without waiting for it to grow. So in one particular analysis, we tried to do just that, learn about those different mutations that are causing resistance within the tuberculosis strains, within the tuberculosis samples. And so we took samples. This was because TB tends to happen really all over the world, and it's it has a fair bit of diversity. It was important that we sample from many parts of the world. And so here we worked with collaborating laboratories across the world to collect samples, and then we wanted to make sure that we have a sample that's balanced between samples that are sensitive and samples that are resistant, so we can make these associations reliably. And then what we did was we looked at how the DNA was changing, and we compared the different samples that we got from the different sites that we studied, and essentially constructed a little evolutionary tree, or like a little life tree of to reconstruct how the DNA changed across the different sites, and looked at where DNA changes happened relative to when resistance was noted. And so this is a little bit of what we did here, was essentially try to identify DNA changes that are happening repeatedly when we observe resistance to happen. And this is one way we can then make associations between the DNA changes and the resistance itself. And what we found was actually similar to what you heard Dr. Kirby present earlier. The antibiotics that we use to treat TB also target frequently the cell wall. And so the mycobacteria, or mycobacterial tuberculosis, this is the kind of bacteria that causes tuberculosis, has a particularly thick and waxy cell membrane. So it's actually many of the antibiotics that we use target that cell membrane. And then when we tried to make associations with these new DNA changes, we found that they occurred in genes that had to do with maintaining the cell wall. So it made sense that we made those associations, and that, in fact, the cell wall machinery was changing in response to drug resistance. And knowing that information allowed us to essentially then move on to construct kind of a model that would take in all the genetic or DNA information, and then would allow us to essentially make predictions about whether or not a particular strain was in fact sensitive to the antibiotics, and we could do that for each of the individual antibiotics that we studied. So this was essentially the results here, that we could make very good predictions for some of the drugs here in green, and not so good for some of the other drugs here in kind of darker orange. And when we looked at the number of different mutations that we found really an inordinate number, really a large number of mutations that need to be identified for us to make good predictions directly without needing to do culture. And so we are following that up using essentially additional samples, larger number of samples, to try to get the performance of our predictions to a level that we could use for patient care. And in addition to this, we are working with tuberculosis, which is in general has limited research funding because it tends to affect relatively poorer nations. We needed to put out all of the results of our analyses out there so people can use them and make predictions and improve upon them. So we designed a web platform that would allow people to input their data and output a prediction of their resistance. And this is a little bit of what the prediction would look like. It would give you a little heat map here with more red indicating a higher probability of resistance, It also gives you an estimate of what the error involves, and you can also hone in exactly on the details as to what mutations actually led to that estimate of resistance. So then you might say, okay, but how would you actually translate that knowledge and those models to actually being able to take care of patients? How would you use that, in a diagnostic way? So I just want to talk a little bit about some of these tests that are currently used for patient care or are currently in development for patient care. So these are tests. This is called the GeneXpert test, which is currently FDA approved and is used actually pretty widely across the world. And what it does is essentially you take in a lung sample from a patient. You are essentially do very minimal processing of that sample. You put it in to this little blue cartridge here, and then essentially put it into this machine. And what that machine does, it essentially looks for certain genetic mutations in the bacteria. And right now, it only looks for a very small number of mutations, and it specifically have to do with one type of drug resistance called, the drug rifampicin. But the hope is that we would then, on the next phase of this, and the companies developing such tools, would allow you to detect resistance to a larger number of antibiotics, because that's actually necessary given the fact that we usually treat tuberculosis with, 4 or more drugs. But in addition to that, there are these other more promising technologies that not only detect 1 or 2 mutations, but also detect hundreds of them. They're so-called arrays, where you have essentially multiple little spots that have different kinds of probes that light up or give a different signal when they identify certain mutations. So these are arrays-based technologies. And the wonderful thing about these tools is that you can get a sample to answer within about 2 hours. So they can really be used almost at the patient's bedside, and the so-called point-of-care test. And in addition to that, these usually cost under $5 a test, which is really incredible. But increasingly, as I mentioned before, we found really hundreds of mutations that are encoding resistance to tuberculosis, in tuberculosis. And so it is more than likely that the capacity of these tests is going to be limited, and they won't necessarily be able to detect the entire span of relevant mutations. And in fact, what we might end up doing is kind of like a sequencing-based test, where essentially you get a sample from a patient and essentially be able to read off the entire genome of the bacteria. And increasingly, there are technologies being developed worldwide that would allow us to do that closer to the patient in a very cheap manner that would also produce high accuracy results. And so one example of this tool is this is a USB sequencer. So this little machine is sort of as big as your finger almost, and is able to generate a whole genome sequence within a few hours. So this is sort of a little bit of a sense of what's coming down. This is definitely not currently used for patient care, but we are optimistic that these kinds of technologies are really going to change the way we diagnose drug resistance, and that's going to be, in fact, necessary for us to shorten that time from when the patient comes in for when we put them on appropriate therapy. So you can see that relative to the current delays of about 1 to 3 months, if you have a sequencing-based technology, was it an array-based or the GeneXpert test, or the whole genome sequencing type technology, that it would take us less than a day. Hopefully, when the patient is right there in front of you, you'd be able to make that determination and then decide right away what the appropriate treatment regimen would involve. And that's it. So thank you so much for listening. So I'd like to introduce next Dr. Michael Gilmore.
Great. Thank you. That's a lot of science to wrap your head around, even if you're in the business. It's a pleasure to be here today, especially in Boston, because Boston has been central to the development of antibiotics and is, I think, going to play a very central role in addressing the antibiotic resistance problem. Let me, you remember that person whose hand you shook at the beginning of this? There's a 90% chance that they're carrying a penicillin-resistant Staph aureus. In the hospital, when you go in, they'll take a swab and whirl it once around your nose, and you'll be tested for something called methicillin-resistant Staph aureus. And about 1/3 of the people are positive for that staph. But that's just one trip around the nose. If you swab the other nooks and crannies of everybody else, you'll find a staph. I guarantee you, everybody has a staph. And 90% of staph now are penicillin resistant. About 1/3 are also methicillin resistant. So antibiotic resistance is real, it's here, and it's on you. And I hope that's the worst that you have. Most of that we can still handle, but it gets much, much worse. As Dr. Hanage pointed out, there are strains now that are pan-resistant to everything, and also, as you heard with the TB, those are becoming extremely antibiotic resistant as well. So it's a real problem. Let me take you back in time before antibiotic resistance was a problem. How long ago, how far back do we have to go before antibiotic resistance was a problem? We have to go back to before the human use of antibiotics. So let me take you back to Boston in 1942. Was anyone in Boston in 1942? No, a few, a few, not many, but maybe a one or two. And in particular, let me take you to November of 1942. And what do you imagine, or what might you remember, was going on back then? Yeah, the U.S. had been in World War II for about 11 months. Remember, December 7th, 1941 was Pearl Harbor, and then so this is about 11 months after that. So the U.S. was really getting involved in the war. A lot of soldiers were going over to Europe. Does anybody know who the #1 football team in the country was in 1942? Boston College. And Boston College, November 28th, played Holy Cross, and they were expected to just wipe the field with Holy Cross and then go on to the Sugar Bowl and be the national champion. But Holy Cross's defensive coach changed the plan and really confused BC, and Holy Cross ended up winning 55-12 and knocked BC out of the Sugar Bowl. So a lot of revelers went to a place called the Coconut Grove. Has anybody heard of the Coconut Grove? Yeah, if you're from Boston, and if you're from Boston until about 1960. After that, a lot of people may not know about it. But the Coconut Grove was a very popular nightclub, and it was able to handle about 600 people. It was a pretty big place. It had several bars and a dance floor and other things. But this night in November, there were 1,000 people there celebrating. Soldiers, revelers from the BC Holy Cross game. In fact, BC planned to have a party there, but ultimately didn't when they lost. But the reason I point that out is because there was a fire, and it was a huge tragedy. Ultimately, just under 500 people were killed in this fire. The fire was a big event for Boston. It was the biggest fire since a fire that happened in Chicago in something like 1903 or something like that. So the biggest fire in 50 years, and I think probably the biggest fire ever since, because ever since then they've changed the building codes. One of the reasons so many people died, obviously there were many more people than the place could accommodate. As I said, it was approved for about 600, but there was near 1,000 there. It didn't have many exits. In fact, it only had one exit that was a revolving door. And when you have a fire, that's obviously not an efficient way to get people out of the building. So a lot of people were trapped inside and a lot of people died. So that night, the injured, those that were burned but still clinging to life, were taken to the Boston area hospitals, and as you know, we've got some of the best. Let me just, 114 people clinging to life were taken to Mass General, and within the first 2 hours, all but 39 were dead. So 39 were taken care of at Mass General. Now, in World War II, it wasn't well known, but penicillin was being developed for the war effort, because in wars, more people die from infections than they die from the injuries. In fact, in the Civil War, 2/3 of the people died from infections. So Dr. Florey, who Dr. Hanaj mentioned, came over, and England couldn't afford, because they were in the middle of holding back the Germans, they couldn't afford to develop penicillin. So Dr. Florey came over and made the case to President Roosevelt and the Defense Department to invest in developing penicillin, which they did. And some of our investigators and physicians at Mass General were involved in this top secret development of penicillin and studying how to use penicillin to treat war injuries. So that night in November of 1942, recognizing that most of these 39 people who made it that far were likely to die from their injuries, the physicians and scientists involved in that research contacted Merck in Rahway, New Jersey, and said, we need all of the penicillin that you've got. Back then, there wasn't much. In fact, so Merck sent all of its penicillin, and it wasn't even purified penicillin. It was just the culture fluids from trying to first steps of making penicillin. So they brought big vats of these culture fluids. It was a 7-hour drive. They had police escorts the whole way. And they were able to save a remarkable number of lives. So the first public use of an antibiotic happened in Boston with the Coconut Grove. So that was the start of it. Now, at that time, effectively 0% of people's Staph aureus were penicillin resistant. And Staph aureus is a main cause of infection after burns. So that solution wouldn't work now. We could try it, but it wouldn't work. In fact, had we tried it as recently as 1960, it wouldn't have worked. So antibiotic resistance in staph to penicillin rose from nothing to about 90% by about 1960. That's how it works. So antibiotics are sort of like where Darwin meets Newton. So for every action that we make biologically with an antibiotic, nature has an equal and opposite reaction. That's the way it works. So let me just take you through a couple of things. So this is a little bit more. It turns out a waiter's match seems to be what started the fire, although that's still being debated in the historical circles. You can see this is what it looked like, and it was said that that match started some of the paper mache palm tree-like decorations, and it went up in flames. The roof was covered in, that's actually a blue cloth. So there was a lot of flammable material, things that now there are building codes that we learned from this very fire. So when you see building codes, it's largely because of this. So, and then does anybody live in the Bay Village area? That's where Coconut Grove was. It's just over, if you know where the public garden is, it's about 2 blocks toward the south end. So there's still a monument there in the sidewalk where the Coconut Grove was. So that's how antibiotics work. Before going forward, let me just say that there are some terrific physicians and scientists in the Boston area who have been integral in this problem ever since that those Mass General days, and I'll touch on a couple of those. But one of the reasons that we have this problem is that antibiotics aren't just used to treat patients. In fact, 2/3 of the antibiotics that are made in the US, or 3/4, are given to animals. And the reason they're given to animals mainly is to promote weight gain. So when you have high capacity animal production facilities, animals don't react well to that and often get sick and are weakened because of the crowding, and they're in close proximity to each other and passing things around. And so people found long ago that if you gave animals some antibiotics in their feed, you could keep that down a little bit and they'd gain weight faster. Okay? So that's what we do. Only about 1/4 are used for humans, and only about half of what's used for humans are actually used in a way that's really useful. So we're squandering this resource. That's the problem. So as I mentioned at the outset, for every pressure we put on bacteria, they push back. The bigger the pressure, the harder they're going to push back. So by not being judicious in what we do, we now are confronted with this problem. Well, why are we feeding antibiotics to animals? Well, we're doing that mainly because antibiotics are made by companies, and companies exist really to make money. And I don't fault them for that. That's why a company exists. You start a company to make a product and to sell the product. If you can sell more of the product, you have more sales. And if you have more sales, you have more revenue. So you have an incentive to find more uses for these antibiotics. As long as we were discovering new antibiotics, we didn't really have to worry about the consequences. We could discover a new one in a few years, and then it really wouldn't be that big of a problem if resistance developed. At least that's how we used to think about it. Antibiotics, on average, have a lifespan of about 5 or 8 years before resistance becomes a real problem, just like penicillin and staph, something like that. So if we can develop an antibiotic every 5 or 8 years, we're okay. We just have to keep doing that. The problem is we're not doing that anymore. And that's, well, let me show you. I skipped over, or maybe it's not in there. Okay. From 1928, when we first came across penicillin, until about the 1960s, we were doing that. We were discovering a new antibiotic every 5 years or so. Since 1980, we haven't really discovered much in the way of new antibiotics at all. We've introduced a few new ones, but they were ones that we knew about before that were simply put on the shelf because they didn't look that attractive for development. We really haven't approved any new classes of antibiotics. We've developed a lot of antibiotics that are modified versions of old antibiotics. So you saw 2nd, 3rd, 4th generations of cephalosporins. You see a lot of tetracycline derivatives. You see a lot of different modified types of antibiotics, but they're all the same class. The problem with that is once you have resistance, it's a much smaller step to resistance to the new class. So we're not developing any new compounds for which we haven't already groomed the soil for resistance. One of the big problems in using antibiotics in animal feed and indiscriminately is that what we're doing, they're doing in the developing world, in some very large, very populous countries like China and India, about 10 times what we're doing. So what we've contributed to resistance, they're now engaged in the same kind of practices that we used. And unfortunately, we don't have the moral high ground to ask for restraint because we haven't set a very good example in that regard. So we haven't been very prudent in the way that we've used it. So, who's going to solve this problem? As I said at the outset, Boston's a very good place to be if you want to be part of the team to solve the problem, and you've seen the three previous speakers. They're all leaders in the field and deeply engaged in coming up with solutions to this. I'll just walk you through a few of them. We've got the Boston Area Antibiotic Resistance Network. That's a working group of people from academics and industry who get together once a year. They're all in the Boston, Cambridge area. Within Boston and Cambridge, we have over 50% of the world's experts, or of the US experts anyway, working in this area. And so we can get together and exchange ideas and find ways to interact and collaborate. And so we do that, and we meet once a year to do that. One of the things that I'm running is called the Harvard-wide Program on Antibiotic Resistance. And so it's a group of investigators from around the Harvard environment, including Mass General, Mass Eye and Ear Infirmary, the med school, the Cambridge campus, and we're working on the problem. There's another Harvard-wide group called the Microbial Sciences Initiative. These are all leaders. Bill is at the Harvard School of Public Health and leading efforts along with Mark Lipsitch at that school, and are deeply involved in looking for solutions. Also, we have a gentleman named Stuart Levy over at Tufts. And Stu, I think, has to be acknowledged in any talk on the subject of antibiotic resistance. He really championed this from the late 60s and has been pushing the case. He started an organization called the Alliance for the Prudent Use of Antibiotics. So we have a terrific nucleus. Some of these efforts are starting to spin out companies. A Harvard scientist named Andy Myers, a chemist, developed new ways to make tetracyclines and new ways to make another class of antibiotics called macrolides. Macrolides include erythromycin, azithromycin, drugs that you might have taken. And so there are two companies based on his technology. There's a woman named Lori Rockme who's looking at ways of killing a microbe called Pseudomonas, and a company called Spiro has started around her technologies. Another innovator in the field is a guy at Northeastern named Kim Lewis, and he's identified some very intriguing new antibiotics and has started a company called Nova Biotics. So a lot of this effort is going in very practical directions to come up with the new antibiotics that we're in sore need of at this point. So what can you do? Let me just give you some practical advice if you're worried about antibiotics. If you have an infection and go to the doctor, ask them if they could culture it. That's a lot of antibiotic use is given empirically without actually testing to see that this antibiotic will work. Whenever you can press for it to make sure the antibiotic is useful for whatever is causing your infection, ask for that. If there are reasons that they can't do it, they'll tell you, but it's worth asking. And don't ask for an antibiotic if the doctor says, well, I'd rather not give you one for what you've got right now. Because a lot of sore throats, a lot of sinusitis, a lot of other things, are not bacterial. They're caused by viruses, and an antibiotic will only attract antibiotic-resistant microbes to you, so that next time you get an infection, it's more likely to be untreatable with that antibiotic. You don't want that. Antimicrobial hand soaps, that's something that a lot of people talk about. Well, it's really unnecessary in daily life. Unless you're taking care of a sick child or something who has something that you don't want to spread around to the rest of the family, that's reasonable. Or if you're handling something like raw chicken. We worked with Consumer Reports and found that 75% of chicken meat from grocery store shelves, and they had a very nice article about this in Consumer Reports, had antibiotic-resistant bacteria on it. So if you're handling those things in the kitchen, an antimicrobial soap is not a terrible idea for that. Or you've heard about MRSA infections in gyms, methicillin-resistant staph infections in gyms. Those have become sort of a point of transmission, so it's not unreasonable to use an antimicrobial soap and washing your hands after that. What can you do to limit the economic incentive for feeding antibiotics to cattle and chickens? Well, you can buy antibiotic-free meat. Might cost you a little bit more, but that's the responsible thing to do. That's what we all should be doing, paying a little bit more for meat and keeping the antibiotics for treating the infections that we get. It's good. You're going to pay one way or the other. And either you're going to pay with an antibiotic-resistant infection or you're going to pay a few dollars more for the meat. So I'd suggest buying the meat. Fish, farmed fish, people are dumping antibiotics into water, into the ocean and lakes when they're farming fish. And that's just basically breeding antibiotic resistance. We don't want that. I'd suggest avoiding it if you can. And then finally, do what you can, if you can, to support research, training, idea exchanges like this. If you are associated with a foundation or a company that's looking for a cause, this, as Dr. Hanich pointed out, is really one of the most pressing public health problems that we have, and it's only going to get worse. And so elevate the consciousness of your coworkers about this problem, people who might be in a position to contribute to it. So with that, I hope all of you escape antibiotic-resistant infections for as long as we all can. And with that, thank you for your attention. And if you would give Dr. Hanich another round of applause for his organizational efforts. And I'll turn it over to Bill to manage the questions.
Thank you very, very, very much, Mike, indeed. And a round of applause for all of the other speakers. And now your hands are tired from all that clapping. I'd like to invite the speakers to go and sit here in front. Now, we don't have a huge amount of time for questions, so I'm going to take them in roughly the order in which they were handed to me, because some of them are really good. And the first one, I think, is a doozy. It is, could you use bacteriophages instead of antibiotics? Now, for those of you who, we may get to this, for those of you who don't know, bacteriophages are the viruses that prey upon bacteria. They very effectively kill bacteria. So presumably, if bacteria are making you sick, we should be able to give a virus which kills the bacteria and not you, and use that as a therapeutic. So I would like to hand that first of all to Dr. Kirby. Would you like to speak to that question?
Yeah, absolutely. So this is something that's in development right now. Bacteriophages tend to be very specific for certain bacteria and specific for certain bacterial strains. So I think what people have found is they have to do a lot of developmental effort, and you may need multiple phages to make sure that you can effectively kill a bacterial pathogen. But there's some evidence in animal models that this actually can work, and I believe there's even some anecdotal evidence in humans suggesting that this may have some promise.
Yes, there was a friend of mine at Yale called Paul Turner earlier this year was able to use a bacteriophage isolated from a lake somewhere in Connecticut to treat successfully an abscess from a patient that was otherwise refractory to treatment with conventional antibiotics. I think that, Mike, would you like to add?
Well, I would say in Boston, has anybody heard of Aerosmith? Has anybody read Sinclair Lewis's Aerosmith from about 1924, something like that? Phages were thought back in the 1920s to be the magic bullet that we could use. And in fact, they were developed for that purpose. And there's an institute in Tbilisi, Georgia, called the Elieva Institute that never got on the antibiotic train and continued to work with phages. I think we got super enthusiastic about antibiotics. I think there's this whole world of phages that we should probably go back to and take a good hard look at. I think it has promise.
Dr. Farrett?
Yeah, I don't have much to add on that, but yes, I agree. I think that the key here is to really be thinking outside of the box towards therapies that are, completely different. So one other thought that's come up on this question of potentially treating infections without the use of antibiotics is potentially therapies that one could use to boost the host's immune system or the human immune system. In some cases, and in the case of TB, there are some such therapies that are currently being investigated. And in addition to that, there are also some thoughts about potentially using even different bacteria. I mean, the question is whether or not we can somehow outcompete the bacteria in our own body and whether or not something like a probiotic would be helpful. This is an interesting idea, very much an idea still, but these are some thoughts that are coming up that are trying to challenge the more traditional use of antibiotics given in emerging drug resistance.
I mean, just to add a little to that and lead us into the next question, a colleague of Maher's was apparently going through British immigration earlier today and was asked what he was doing there. And he said, I'm giving a talk about antibiotic resistance. And the guy on the border said, really, why do you use bacteriophages? Which was quite impressive. But one of the answers that he was thinking about giving is that they're very strain specific. So just like, think about natural selection. We have very good immune systems in order to throw off the things which infect us. Bacteria are very good at avoiding bacteriophages. There's a very strong selective pressure for that. And so every virus is typically only good at fighting or killing one particular strain of bacteria. And if you don't know what you have, then it's a guess. Which leads us into the next question, which is about empiric or best guess treatment. As several people mentioned, when we're treating disease, we often make a guess as to what's there. And so we take what's like a broad spectrum antibiotic. And boiling down the question, could there be a sort of comment on the sensitivity / specificity of such guesses? Do you have anything to say about the practice of broad spectrum therapies as opposed to more targeted things? And I think I'll leave that to you, Maher.
Sure. So I think that it would obviously depend on the kind of infection you're talking about. So there are a lot that we know about the so-called epidemiology, meaning the kinds of infections that cause certain symptoms. We don't know very much, given that each type of bacteria has been found to have a range of sensitive to resistant, whether or not that particular, say, bacteria is going to be in fact resistant. But so we have, I guess is what I'm saying, is that you have some idea, but you're not definite about the range of infections. For example, in the case of, say, strep throat is almost, universally going to be penicillin sensitive, so you can reliably treat with a very narrow spectrum antibiotic. But for certain other infections like pneumonia, say, there's really a variety of different infections that you use. So we are often treating so-called empirically. And the success rate of broad spectrum antibiotics for the most part is very high. But I think increasingly we're seeing that in fact are even in some cases, again, currently thankfully rare, that they failed these broad-spectrum antibiotics. I think ideally, which sort of goes back to Dr. Gilmore's recommendation, when you can get more information about what the infection is, meaning through culture, if you can, that would be ideal, because that would identify exactly the infection that you have, and it would allow that, the further testing on whether or not it's sensitive to antibiotics. And that would be, you would be, essentially not driving blind, hoping for the best.
Anybody want to add to that? I'm slightly surprised you didn't mention genomes and DNA, because if you could use DNA to identify what we were susceptible or resistant to, problem solved. Anybody?
Go ahead, James.
Yeah, sure. So if you have a bloodstream infection, for example, with an organism which was mentioned before, Pseudomonas, every 24-hour delay in getting patients on the right therapy confers about a 10% increase in mortality. So the stakes are high, and with emergence of antimicrobial resistance, our guesses for empiric therapy, even if we give broad spectrum, may be wrong. So I think it underlies our need to have more rapid diagnostics so we can figure out very quickly, because there are so many different possibilities now in terms of what might be remaining. So we need to figure out very quickly and have the diagnostics that can tell us as soon as possible what an organism is susceptible to.
I would just add that we use clinically what we have. And we have broad spectrum drugs, most of which can be taken orally. And the reason we have those is because those are drugs that could be used for the greatest spectrum of conditions. So if you're a company, again, making a drug, that's what you want. You want to be able to get a lot of it out there and do it. If you have a very narrowly focused drug, you're only going to sell a little bit of it to just those people. And it's not going to work for a lot of other things. We really need broad spectrum drugs that work because in the first minutes, as James said, we don't know precisely what's causing it, but we want to stop it as quickly as we can. Because every minute that you don't treat a serious infection, mortality goes up. So you want to give a drug that'll work before you even have information. So broad spectrum drugs are really important for that, but again, we should use them very sparingly. I would have to say, when you cross the 50-year-old age level, all of a sudden, you get covered for a visit to a periodontist. And so your dentist almost always will refer you to a periodontist because now it's paid for. And I don't mean to malign any practice of medicine, but I was advised to take 2 antibiotics, metronidazole and ciprofloxacin, for a condition that's not very severe and that I'm not really sure that I had. And that really is a profound dose of antibiotics to anybody. And I would say that should only be reserved for the worst infections that I might have, but that's not what's happening in practice. So I think we need to heighten people's awareness that, okay, if I don't really need antibiotics that badly, I shouldn't take them.
That was an excellent point, which I would echo. Unfortunately, we are running out of time, and I can see some people are unfortunately having to shuffle out the back already. So in the interest of that, I have one more question, which I think might be directed at Mike, but I would like to get a question from everybody. Crisply, please. What's bad about antimicrobial soap?
I would just very quickly say that we need the normal bacteria on our skin. We need the normal bacteria in our guts for our own health. When we remove the bacteria on our skin with an antimicrobial soap, then that opens the door to ones that aren't as benign. So it's those benign bacteria that we carry around in health that we need to preserve.
Better the Staphylococcus, you know.
Yeah, that's exactly right.
Does anybody else have anything to add to that? Okay, in which case, and all that remains me is to thank Dr. Gilmore, Dr. Kirby, Dr. Farhat. I want to thank all of you. I want to thank Belinda and Susan who managed to organize this, and thanks all for coming. Sorry if I didn't get to your question.